Hello everyone, welcome to the new lecture on medicinal chemistry. Today we are going to explore one of the most fascinating concepts of medicinal chemistry drug design, that is the bioisosterism. So in today's lecture we will explore what is bioisosterism, why small structural changes can modify the drug potency, efficacy and safety, and why small structural changes are highly important. We will explore the different types of bioisosterism. We will explore the applications of bioisosterism in the drug design and medicinal chemistry, and we will check the effect of different small functional moieties on the activity of drug molecules. And we will also check out the real world example of the already FDA approved drug which are synthesized by the concept of bioisosteres. So let's dive deep into this concept. So, have you ever imagined that how small chemical changes could turn the toxic drug molecule into a life-saving medicine? Or why and how medicinal chemists modify these small drug molecules and convert them into the safer and more effective drug? The answer is the bioisosterism. As this diagram shows that in the drug discovery process, we have the molecules which are more toxic, but by making certain modification, its therapeutic index can be changed and we can make a safer drug from it. So the story behind all this concept is the bioisosterism. Let's suppose, consider yourself as a medicinal chemist. If you are a medicinal chemist and you have explored a lead molecule, but the problem is your lead drug molecule have still some issues with their pharmacokinetic pharmacodynamic, means it has some metabolism issues, and it has also the issues with the binding with the receptor. So that means it is still not ready to be converted or synthesized into the drug molecule. So for this purpose, this lead molecule need to be modified. So that process is known as the lead optimization. So the lead optimization is a process in which we modify the lead molecule to produce a target clinically safe, effective and potent commercially available drug. So how lead modification take place, and the concept behind all these is the bioisosterism. So now let's dive into the history roadmap for the bioisosterism concept. The bioisosterism concept evolved from the isostere concept, which was introduced by the scientist Langmuir in 1990. Later on, another scientist, Grimm, supported the Langmuir isostere concept, and he added the further point of his hydride displacement law in 1925. And in 1932, Allen Ayer further extended their isosteric concept and make it more effective to lead a foundation for the bioisosterism concept. So the real breakthrough for the bioisosterism comes in 1951 when Friedman introduced the word or term bioisosterism. He also gives the relationship between the chemical structure and the biological activity of the drug. As in this diagram, you may observe this is the amide functional group and this is the ester functional group. They have the similar biological properties as well as. And in 1991, Berger further supported and extended his bioisosterism concept by adding some few points of the isostere and the steric concept. So now let's check out the detailed theory behind the scientist concept. Like in Langmuir in 1919, he observed that there are the certain co-molecular isostere which are the molecule or group of atom which have the same number of electron, and on that basis they have the similar physicochemical properties. So as in this table, this is the table from the Langmuir isostere. In this you may observe hydrogen, helium, lithium have the 2 valence electron, and similarly these underlined like nitrogen, carbon monoxide and night have the 14 valence electron. So this is basically the table or the group of atoms which are arranged by Langmuir as the isosteres, and he observed that they beside having the similar valence electron, they also possess the similar physicochemical properties. Later on in 1925, Grimm further expanded the idea of isostere, and he included the substituting atom with different charges could produce the similar bonding characteristics. Like he observed that the fluorine, hydroxyl, amino, and the CH3, these are the group on which the hydrogen is displaced, and they have the similar bonding characteristics, and they can be used as the displacement or modification in the drug design. And later on, Elmner further extended the Grimm classification, and he defined that the isosteres are the atom, ion, or molecule in which the peripheral layer, which is the outermost electron, are the identical. So in the Elmner classification, this is the diagram. You may observe that these are the group 5 atom. These are basically you may observe that the different include the different atom from the periodic table, and also the atom or group of atom which are substituted with the hydrogen, like nitrogen and phosphorus are the group 5 atom, and it also includes CH and SiH and the strontium. These are displaced with the hydrogen. So the purpose of grouping them together is because they have the similar bonding characteristic, and they can be replaced in the certain site in the drug design. The real ground breakthrough in the Bioisostere concept was given by the Friedman in 1951, who coined the term Bioisosterism. He observed that bioisosteres are a group of atoms or molecules that are structurally similar, and they show the same type of biological activity. And further, in 1991, Berger further extended or supported his concept, and he said that the molecule or group of atoms which possess the similar molecular shape, volume, and approximately same distribution of electron, and they have the similar physical properties and biological properties. So in this diagram, you may observe this is the amide functional group, and this is the ester functional group. These both groups have the similar shape, similar electronic distribution, and the similar physical properties. So all these such functional group which have these kind of similar characteristic, molecular shape, electronic distribution, and physical properties, are said to be the real bioisosteres. Let's check out the applications of bioisosteres in medicinal chemistry. Bioisosteres, we have observed that this concept is evolved from the isosteres, which have the similar physical and chemical properties such as size and their electronic configuration, electronegativity values, and so on. So these are the functional group which could have the kind of biological activity. So in drug design, these kind of isostere can be replaced with the similar isosteric which we could possess the biological activity. So the size and shape represent the hybridization arrangement of atom bond angle and bond length among the atoms of functional group, and the electronic arrangement like the inductive mesomeric effect, polarizability of their functional group, the charge distribution, electronegative or electropositive functional group present in the drug structure, which will ultimately affect the water solubility and the lipophilicity values or pharmacokinetic properties of the drug, and their permeability across the membrane, and they will ultimately affect the hydrogen bonding and the pharmacokinetic and pharmacodynamic, that is the affinity to bind with the target of the drug molecule. So all these factors are modified when the drug is replaced with their alternative bioisosteres. So ultimately, by the application of the bioisostere, new drug molecule design will be improved in their stability, potency, selectivity. So bioisosteres in medicinal chemistry will be helpful in a design of drug molecule which have the good stability and which have the improved potency and higher selectivity with its target, and they have the more optimum pharmacokinetic properties, and they are good absorption and the high membrane permeability, optimum pharmacokinetic profile. They have the good binding affinity and their lower side effects and the higher safety profile while maintaining their biological activities. So these are the most important application or advantages of bioisosteres in the medicinal chemistry. Classification of bioisosteres. Bioisosteres is mainly classified into the two main classes, classical bioisosteres and non-classical bioisosteres. Classical bioisosteres have the further subtypes monovalent atom, divalent atom, tetravalent, trivalent, and ring equivalent. Non-classical bioisosteres are of three types: cyclic, non-cyclic, functional group, and the retro-bioisosterism. So now let's check out the classical bioisostere. Classical bioisostere are those which truly obeys the Berger steric and electronic definition. Means they have the similarities in shape and electronic configuration of atoms and group of atoms. So those bioisosteres, those functional group which are to be replaced must have the similar electronic configuration and the steric properties. So these are more helpful in the design of the drug molecule, which will enhance the affinity or the drug binding affinity of with the receptor or with their target organ. The types are the monovalent atom. So among monovalent, it is the fluorine, which can be replaced with the hydrogen, and the OH replaced with the NH. Fluorine, OH, NH or CH3 can be replaced for the hydrogen, SH and OH. So these are the hydroxyl amino, methyl for H. These all are the example of monovalent atom or monovalent groups which are considered as the classical bioisosteres. So now let's check out the real FDA approved example of drug which are based on the bioisosteric concept. The example of the drug is the uracil and 5-fluorouracil. Uracil, we know that it is the one of the nitrogenous bases present in the DNA. You may observe that at fifth position it has the hydrogen, but when this hydrogen is replaced with the fluorine, it is converted into a drug molecule that is the 5-fluorouracil. And another is the hypoxanthine. Hypoxanthine is in this you may observe that it has the hydroxyl functional group, but when this O of oxygen atom of hydroxyl is replaced with the sulfur, then it is converted into a drug that is the 6-mercaptopurine. Why this modification is done? Because this is the cancer cell, and you know that the cancer cell division and growth requires the DNA, and for the DNA the uracil base is important. So once the hydrogen is replaced with the fluorine and the drug, it is converted into the 5-fluorouracil. Now this 5-fluorouracil will act as the anti-cancer drug, and it will act as a cytotoxic to these cancer cells. So the growth of cancer cells will be diminished, and ultimately it will kill the cancer cells, and it will be effective and potent against the cancer cell in comparison to the uracil, which was promoting the growth of cancer cells. In the same way, the replacement of the sulfur with the oxygen in the hypoxanthine lead to the mercaptopurine, which is the also an anti-cancer drug used in the treatment of leukemia. Divalent atom are groups. So the examples are the carbon sulfide, carbon monoxide, or the carbon NH, and the carbon-carbon double bond. So here are the example of the true drugs which are based on the divalent classical bioisosteres, procaine and the procaine amide. In their structure, you may observe in procaine we have the ester functional group at the para position, and when this ester functional group is bioisosterically replaced with the amide functional moiety, then it will changes entirely the drug molecule from one category to other, because it is converting the procaine into the procaine amide drug, and both have the different pharmacological property, because our procaine is used as a local anesthetic and the procaine amide is a anti-arrhythmic drug. So we have learned that the Bioisosteric concept is also useful in developing or designing a new category of the drug molecule from already existing. So the trivalent atom are groups, example are the phosphorus, strontium, nitrogen, CH, and the tetravalent atom include these different group phosphorus, strontium. So we will not go in detail of these, but then we are uh, checking out another example of classical bioisostere drug. So this is the caffeine, which is the purine base, and in this you may observe that it has the methyl group, and when this is demethylated, it is converted into a drug molecule, which is the theophylline. Theophylline is one of the bronchodilator which is used in the asthma treatment. So this is the these structure shows a classical bioisostere relationship between the xanthine derivative caffeine and theophylline. Another example of classical bioisostere is the development or design of the NSAID drug. So the salicylic acid derivative, this is the structure of salicylic acid. In this you may observe here we have the phenolic or hydroxyl functional group. When this is replaced with the NH2 functional group, it will be converted into a new class of drug molecule, that is the anthranilic acid. So the salicylic acid or anthranilic acid are basically the foundation for the development of the other derivative of or other generation of the NSAID drug category. So when salicylic acid, this hydroxyl functional group is acetylated, it is converted into the acetyl salicylic acid, which is our aspirin, one of the famous well-known NSAID drug molecule. And if this NH2 group of anthranilic acid is further modified with the heterocyclic ring, it will get converted into the mefenamic acid. So what we have observed that the classical bioisostere can also be helpful in designing of the more potent, highly safer and effective NSAID drug molecule. Now let's check out the non-classical bioisosteres. Non-classical bioisosteres are those bioisosteres which do not obey the steric and electronic definition of classic isosteres. Means they do not have the same number of atom or replacements. Means it is not important for them to have the same electronic distribution or to have the same steric properties. So they could have any one of the following characteristic, like they could have maybe same electronic properties, same physicochemical or a steric or a spatial arrangement or functional moiety, but it is not mandatory for them to possess all these characteristics. So they are not the true Burger bioisosteres. So non-classical bioisosteres, in this there are the different examples. Cyclic versus non-cyclic means the replacement of the cyclic ring in a drug with the non-cyclic side chain, and we can observe a different drug example, like a well-known example of non-cyclic ring opening is the diethylbistrol, that is the one of the famous synthetic derivative of estrogen steroid hormone. You can check my estrogen SAR lecture on this topic and you may observe the structure of this estrogen derivative, that how ring opening is occurring and how this derivative is synthesized. The other example we will check the lidocaine, which is the local anesthetic and which is used which is synthesized from the mepivacaine. So this is the non-classical bioisostere example of the development of the lidocaine from the mepivacaine. The lidocaine synthesis from mepivacaine is based on the concept of ring opening. The ring opening, because in this mepivacaine you may observe we have the terminal cyclohexane ring at this position, and when ring opening is caused, we have here a side chain. So this ring opening enhanced the drug stability and it converted into a more potent local anesthetic agent, that is the lidocaine. So carboxylic acid isosteres. The carboxylic acid is a functional group which is present in the many drug molecule, but it has some limitations like low oral bioavailability, rapid metabolism, so they must be overcome by the bioisosteric replacement with the certain functional moieties like tetrazole, SAL, sulfonamide, hydroxamic acid, sulfonic acid, and so on. So these isosteric will maintain the hydrogen bonding and electrostatic interaction of carboxylic acid with the receptor, and at the same time they will improve their pharmacokinetic profile. So these are the different functional group which can be used in place of carboxylic acid as a bioisostere, like tetrazole, hydroxamic acid, acetylcinamide, hydroxamide, acyl sulfonamide, sulfonic acid, phosphonamide. These all are example of carboxylic acid bioisosteres. This is the example of drug which is based on the replacement of carboxylic acid group with the tetrazole. The example is the Losartan. This is the one of the alpha-2 receptor blocker which is used in the treatment of hypertension. So when the carboxylic acid group was replaced with the tetrazole, it is converted into the more potent drug molecule. Another example of carboxylic acid bioisosteric replacement is the PABA, para-aminobenzoic acid, in which carboxylic acid is replaced with the sulfonamide functional group, will lead to a sulfanilamide, which is one of the classical example of antibacterial drug. And sulfanilamide is the structural analog of PABA in sense of the electronic, conformational, and physicochemical properties. But therapeutically it is totally opposite of the PABA, as PABA is required for the bacterial growth. It is a major precursor for the bacterial growth and metabolism, and sulfanilamide acts as the antibacterial drug molecule. Let's check out the other example of the different bond replacement, like the amide bond isosteres. Amide functional group is present in the most of the drug molecule. It can sometimes also be replaced with its bioisosteric equivalent like 1,2,4-oxadiazole, 1,3,4-oxadiazole, or 1,2,4-oxadiazole. The purpose of replacing the amide bond functional moiety is to prevent the enzymatic breakdown, and this concept is most importantly used in the protease inhibitors of the HIV therapy. Another functional group example is the aromatic ring isosteres. Aromatic ring benzene have the high lipophilicity and solubility issues, so it can be replaced with their other bioisosteric equivalent like pyridine, which is the heteroaromatic ring, and it can be replaced with the saturated ring like cyclohexane, or acyclic like alkene. These all saturated hetero are the acyclic equivalent of benzene have the electronic distribution properties which will modulate the solubility issues of the benzene aromatic ring, and they will lead to a certain flexibility in the conformation of the drug molecule, which will modify the binding affinity of the drug with its target and will lead to a more effective or potent drug molecule. Heterocyclic ring equivalent include the heterocyclic replacement of the pyrazole with the imidazole and the benzene for the pyridine and the other rings. So these are the different rings. Benzene can be substituted with the thiophene, which is the 5-member ring with the sulfur heteroatom. Furan can be replaced with the thiophene. Furan is having the 5-member with the oxygen and the thiophene with the sulfur, and benzene can also be replaced with the nitrogen. So these are the different examples of the functional group which are also included in the Bioisosterism concept. So to summarize this lecture, I would say the Bioisosterism we have observed is a game changer in the drug design and drug discovery process, as we have observed that how it is important in improving the drug pharmacokinetic, pharmacodynamic stability and safety, and it helps us in designing the better and safer drug, for the example different anti-cancer drug, different antibiotic, CNS medication, and the other different classes of the drug. So thank you so much. I hope you have understand a lot. Thank you.